Hi, everybody. Welcome to MetaXpress's February webinar on taking analysis into your own hands, creating modules using MetaXpress software 5.0. I see that many of the names I see on the registrant are people who are customers and who have come to previous webinars. Welcome back. I'm the product manager for cellular imaging at MetaXpress, and Paula, a longtime application and support scientist, will be co-giving this webinar today. If you need help or technical support, there's an 800 number that will work to get help directly from the WebEx. And also, I'm going to go to a slide that explains how you can just send Q&A to both us and to WebEx. In the meantime, just to remind you, so I'm the product manager, and I'll be introducing the MetaXpress high content screening systems and what's new with the product. And then Paula will be going through some examples and a live demo of creating modules using the new software. And we will summarize at the end with some Q&A that you can send as we go. So please submit questions at any time during the webinar. We'll compile those and ask those back to the panelists at the end. Press on to submit a Q&A. Press on the Q&A button on the toolbar to expand the Q&A window, and then you'll see this dialog, and then you can type your question in there. Please choose all panelists so that we all will see those questions, and then hit the send button and for us to be able to view those questions. So to remind those people not familiar with our product, what our offering is, we have a complete solution for imaging at here at MetaXpress. On the top left is our wide field high content screening system, the MetaXpress Micro. We also have a confocal system, the MetaXpress Ultra. And we've recently introduced an ability to import third-party images into our data management solution. So there are two systems, the wide field, the confocal, and if you have third-party instruments generating data, they can all be imported into our data management offering, MDC Store solution. This allows for both image analysis and converting those images into numbers to be done with MetaXpress and our MetaXpress PowerCore software option. In addition, all of that data and data management in one location can now be tied together for hit selection with the MetaXpress software. We've talked to many customers, and they're encountering they repeat the same type of challenges that they're encountering in their screening centers. They want more capacity to acquire and analyze screens. They have a rapidly expanding application space that needs to be addressed. They want to cut down wait time for screen visualization and quality assurance. And they want to integrate with existing imaging modalities or alternative imaging modalities. So we're trying to bring tools to address these challenges to the market. So I'm going to start with a little bit about what's new in MetaXpress. That will introduce what Paula will show you later. So there's hundreds of high content applications out there, all the way from apoptosis to zebrafish. We like to think that we can address these applications from A to Z. And there's a long list of things on the left that need addressing that happen in high content screening. You need to approach those complex applications. You need a comprehensive toolbox for your imaging needs. And there are very well-defined applications. You also need something very flexible for those not well-defined applications. So there's an analysis continuum. And an analysis continuum starts with robust acquisition with some simple case-centric or customizable on-the-fly decision-making tools, which we have. For those well-defined applications, we have application modules which give you a turnkey for common applications. For the ultimate flexibility, we have journals which are macros that allow for advanced and interactive applications. But there's a little bit of a possibly a gap in there between those two extremes, and that's where our new ability to create custom modules that will bridge that gap. And once a custom module is created, it allows you to make that now a turnkey application. So once it's created, there's very few parameters that need to be altered for that to now become a turnkey application. So why do people want to go to custom modules? You can do a lot with custom modules. It gives you an expanded set of tools for measuring transmitted light images, and Paul will go through an example of that. You can do many morphometric classifiers, shape analysis. You can find objects within objects. And once created, custom modules are modules. They can be shared. You only need to modify a few parameters to use it on a different set of images. It can be run through our parallel processing option, MetaXpress PowerCore, just like a CAN application module, and it's seamlessly integrated into our database once it's been saved. So here's an example of a pretty good-looking image that was taken without any fluorescent fluorophore, and we can use this new custom module to segment those images. Paul is going to go through some example with much lower contrast, higher, more challenging images to show you the power of the new tool. You have an all-in-one interface to create this tool. At the top of the custom module editor is a place to choose your segmentation tools to be able to find your objects. You can use existing application module as maps to find objects. You can modify your objects. You can modify your images. As you choose and select steps, those steps show up as cards on the left, okay, with a numerical value. There's 1, 2, 3, 4. Those are 4 steps. And those 4 steps correspond to a thumbnail film strip at the bottom to see what you're doing as you progress through your image analysis routine. And if you need to go modify one of those steps, you just click on the image or click on the card to modify that step in the module. Here's an example of a custom module created in Metamorph, which is our sister product for research imaging. Again, it has the same look and feel. All the steps corresponding to segmentation are viewed at once. The module that was created in Metamorph can now be imported into MetaXpress and reused there, and that module can be run through MetaXpress PowerCore. Now, this example is the ability to find cells. The blue is the nuclei, the cells are the cytoplasm, and then find the intensity of the plasma membrane to find intensity and shape that has to do with the cell's plasma membrane. So not only is the custom module there in MetaXpress, it's there in Metamorph. You can do some assay development on your microscope and reuse that module in MetaXpress in a high content stream assay. I mentioned PowerCore a couple of times. So this is our tool for removing analysis bottlenecks that might occur with image processing. It's compatible with both custom and turnkey modules. It is a MetaXpress software option. It drives your analysis to be faster than image acquisition, and you license the number of cores you need to run across to drive that analysis down. Somewhere between 4 and 16 cores is when you will be well above acquisition speed, and your analysis is going to be taking minutes. Also, I wanted to just remind you of a couple of other things that are new. I mentioned that you can import third-party images into our tool. This is a new tool that allows you to automate and schedule import and export of images from MDC Store. There's a number of formats that are compatible. A number of TIFF formats are easy, OME files, and there's a list of file formats that can be imported. And it automates the transfer images across sites or departments. It runs through a web browser, it connects to the MDC Store database, and you can schedule and automate the movement of these images into and out of our database. Additionally, I want to mention some improvements on the hardware. So many of you know we have the Excel system and we have the standard system, and the top left compares the field of view of those two systems. The Excel is a little bit over 3 times the field of view of a normal standard high-content screening system. And the big box represents a 3D4 well, and the small green box is the field of view with a standard camera. With the new XL, the light engine was changed and the detector was changed so that with a 4X objective you can capture more than a 3D4 well. It gives you 3 times the number of objects. If you want to do low resolution imaging, you can get 1 well in a single image. And if you have large objects or high resolution, you can collect less images for those assays. Additionally, the XL standard, the standard configuration of the XL system has a solid-state light source that gives you on-demand illumination, eliminates mechanical shutters, and reduces support requirements. Additionally, we will work with you to implement other long-life light sources that have been validated with our system. This gives you the flexibility to address a variety of applications and pick a configuration that works for you. This now enables UV, IR, ratiometric imaging, excitation switching. So speak to us about what you need if our standard system or our standard Excel system are not adequate. So now with MetaXpress software, we are driving for fast and simpler. So we want to alleviate your day-to-day challenges to be able to run more screens, grow with your expanding applications, perform hit selection faster, speed up analysis. We pride ourselves on maintaining quality images, and now we have the tools to also integrate with existing imaging modalities. On that note, I will pass the presentation over to Paula, who will go through some examples of using the new software tools. Paula?
Hopefully everyone can see my screen now. So thank you, Grisha, for the introduction. So I'm here to give a little bit more of a specific introduction to the Custom Module Editor, or CME, and its use in a few specific applications. So as Grisha mentioned, with the CME, we are now able to simplify doing custom analysis in order to do certain tasks that are pretty common requests, and until recently we've had to address through journals or our custom macros. So some of these include running multiple modules and combining the results together, running one segmentation but then getting measurements across multiple wavelengths, or segmentation of transmitted light images. So I'm going to show you some examples of these live in the software today so you can see exactly how quick and easy it is to set up a custom analysis. So I have 3 examples that I'm going to go through. One is a bead phagocytosis assay, and then also a bright field cell proliferation assay and a cell migration assay. So we're going to go ahead and start with the bead phagocytosis assay. So phagocytosis is a common process that's very important for the immune system, for immune cells to engulf bacteria and other particles, as well as dying cells. And in a typical assay, you'll have a particle that's coated with a ligand and also coated with some sort of fluorophore. And if the cell is able to phagocytose, you will see it engulf the particle, and you'll see it appear inside the cell. So for this particular assay, we have cells that had a bead supplied with the appropriate ligands in 2 colors, and a DAPI stain for nuclear identification. And the cells were treated either with a DMSO control or with a compound known as cytochalasin D, which inhibits actin polymerization, so thus inhibits the phagocytosis. And I apologize, I don't know what the exact ligands are on these samples because I didn't create the samples myself. And the goal that we're trying to read out for this particular assay is an overall count of the cells, and then for each cell, how many green-only beads are phagocytosed, and then how many double-stained beads are phagocytosed. Okay, so just so you can see some example images, in the blue, or the DAPI, we have the nuclear stain, In green and red, the FITC and Texas red channels, we have beads, and when you overlay them, you will see that some of the beads are green only, some of the beads are red only, and some of the beads have both fluorophores. Okay. And when we ran the analysis, you can just see quickly that with the treatment, we see a very distinct phenotype in both measurements of the green-only beads per cell, as well as the green-red co-stain beads per cell. So the strategy that we're going to go through is first find all the nuclei, and then expand the nuclei to cover the cell area. Then separately, we need to find all the green beads, and find all the red beads, and then restrict that count to those just inside the cell area that we're looking for, and identify how many have the green and red stain, and how many are green only. So at this point, I'm going to go ahead and switch over to the software. So here you can see the plate showing in the color overlay. And if I go to the Run Analysis tab, you'll see I have my normal list of modules. If I have created any custom modules, they will be listed down here at the bottom. I do have one actually already created for this, so if I wanted to edit it, I can click on Configure Custom Module. But for the purposes of showing how easy it is to put together that custom analysis, we're going to go ahead and create a new one from scratch. So I'm going to click on Create Custom Module, and this will open the Custom Module Editor. And it takes just a few seconds for this to open. Okay. So I apologize if this is a little bit small. We wanted to make sure that you could see all the components rather than having you scroll around on the screen. So what you're seeing now are all my input images, the DAPI, FITC, and Texas thread images from the site that I was looking at. And here will be all my steps for my segmentation, and then there's a separate tab for measure when I'm ready to set up my measurements. So I'm going to start with the segmentation. And the first thing that I want to do is find my nuclei. So I'm going to go to the Find Objects collection here. I have a few choices. And in general, we find for nuclei, either auto-find blobs or find blobs works quite well. Okay. So I have my find blobs here, and I need to select my source image. So I'm going to set this to DAPI, and it automatically updates to DAPI here. Now, you may or may not be able to read this on the screen. It has a place I can enter an approximate minimum or maximum width and an intensity above local background. Now, for those of you who are used to using our CAN modules, these numbers may sound familiar, but there's an added tool that will make this a little bit easier for us to work with. I'm just turning up the brightness here for people to see. This button is a click to find, and so if I activate this, I can now just click on various nuclei in the image, and you can see they are being selected on the right. And my values for the width and intensity are being updated as I click on these. Okay. So once I think I've gotten a representative sampling of the different nuclei I want to detect, I can just go ahead and name my result, which in this case will be nuclei, and click Apply. And you will see how it's found all of those nuclei here on the right. And you can either look at this side-by-side view, or if you want to see them overlaid, you can go to the Split View option here, and then this line becomes a divider that I can move left and right. Okay. All right. So if if I'm happy with my nuclear segmentation, I can go ahead and go to the next step, which is to expand out the nuclei to create the whole cell area. And so now I want to work with my nuclear mask, so I'm going to go to the Modify Objects. This is modifying an object mask that you've created. And you'll see we have options to grow and shrink objects. There's also grow objects without touching. And this is the one I want, so that I can expand my nuclei, but then keep each expanded nucleus as a separate object. So my source will be my nuclei, automatically updated with my name here. And then I don't want ultimate growth, and I'm going to grow by a fixed amount. And I'll give this a name. And you can see as you try different settings how much it grows. That's 3 pixels. And I can do a larger amount. And this to me looks about right. It's about 15 pixels. It's about the typical cell area for these cells. Okay. So now I'm ready to look for my beads. And so I'm going to go back to my Find Objects collection here. And the beads are very nice and uniformly round. So for this, I can use the Find Round Objects tool. And it pulled up the Texas red image, so that's fine. I'm going to go ahead and select that. And I'm going to, again, use the Click to Find, and I'll click on individual beads. Okay, and it's updated my settings here. And I'll just name these red beads. And click Apply. Okay. And I'm going to do the same thing for the green beads. Find round objects. This I'm going to select a 50 image to start. Click to find. And actually, if you need to zoom in, you can use these zoom buttons, or you can also use your scroll wheel. So I'm going to go ahead and just click on some of these beads. And I'll name this green bead. And click Apply. And then I can zoom out and make sure that it's found things appropriately. Okay. And if I need to at this point, I can eliminate clumps like this by adding in a filter. There's a filter mask option in here. I'm not going to bother because I'm just going to include everything just to keep it simple. But it's very quick to add in. So now I basically need to identify which of the beads have both green and red and which ones are just green. So for that, I'm going to use the tools Keep Marked Objects and Remove Marked Objects. So for Keep Marked Objects, my object source will be the green beads, and the marker source will be the red beads, and the results will be my green and red beads. Okay. So you can see only the beads that appeared in both the green and the red are now showing in this green-red mask. And now I'm going to select the Remove Marked Objects. And my object source will be my green beads. And my marker source will be the red beads. And now this will be the green-only beads. Okay? And now you will see that all the beads that had green but do not have red are what are appearing in my final mask. So I've created a cell mask, and I've created a mask of the green-only beads and the mask of the red-only beads. The green-red double-chain beads. Okay, so those are all the masks that I needed to generate for my module. So now I'm ready to go ahead and create my measurements. It's Usually by default we'll save the overlay, the analysis overlay, which you usually want to see when you're reviewing your results, so we'll go ahead and leave that on. What I need to do is to select my main mask here for my cells, and that's the expanded cell area. And if I want to get any measurements, intensity measurements, etc., I can do that here by selecting an image. In this case, I don't need any of the measurements for this particular area, the expanded cell area, so I'm going to deselect all of these. But you can see from the list that there's quite a number of different types of measurements available, including size, intensity, and morphological measurements. So now what I want to do is measure different features within that cell area. So I'm going to add a feature group. And first I want to measure my nuclei, and I'm going to measure that from the DAPI. Okay. And all I really need here is my nuclear area. I might want my average nuclear intensity. And I should only get one nucleus per cell in this example, but I can add that on. And you can see that I can rename any of the measurements that I'm selecting, which is nice because I can customize it for my particular assay and makes it much more clear in the final readout. Okay, so now I'm going to add another feature group, and this will be for my green-red beads. And actually, the only measurement that I am particularly concerned by here is the count. So I'm just going to select the feature count at the bottom and name this my green-red bead count. And then I'll add another feature group here, green-only beads. And again, the only measurement I'm interested in here is the count. Features count, I'll name this green-only bead count. Okay. All right. And now I can go ahead and click Apply to test that. And this will show me on the right my cell-by-cell data. I'm going to zoom in on some of these results, and I can click on a cell. And if I look at this, it'll tell me this particular cell had 1 green-red bead and 6 green-only beads. And if I toggle the overlay, I can see that looks correct. Okay. So if I'm happy with my analysis at this point, I can go ahead and name and save my module. If I wanted to tweak any of the settings, I can just easily go back to the segments tab and adjust any of the parameters and test the results just as easily. Okay, so then I can go ahead and save this. And you'll see on the normal MetaXpress screen that it's added the custom module successfully. So I'll click OK, and I can go ahead and exit out of the CME. And now you'll see it selected in the list. It's been added to my list here. And I can go ahead and select some wells to run it on. And you'll see it's running through. It runs nice and quickly, much quicker than most of the custom journals that you might have tried in the past. And this is not running PowerCore, so with PowerCore it would run even faster. This is with 4 sites per well. Okay. Okay, so now you can see, oops, here we go. I can select my measurements. You'll see all the measurements that I entered. So my most concern is the green-only bead count per cell, and you can see the difference in the left, the cytochalasin bead treated samples as well as the control samples here. There's definitely a big difference in the output. And if I look at my results, I can see the overlay here, and I can see it identifying those beads nicely. And I can also look at my green-red bead counts and see that that goes down just as I would expect. Okay, so that's the phagocytosis assay. So hopefully that was reasonably straightforward. I'm going to go ahead and go back to my presentation now. So just to remind you of what we did in this custom module, we found the nuclei, expanded the nuclei to create my cell area. I found all the green beads. I found all the red beads. And then I looked at which of those overlay to find the double-stained green and red beads inside the cell region, and then also which were the green-only beads inside the cell region. And then I set up my measurements and ran the module. Okay. So the next example I'm going to show you is a cell proliferation assay, which was done in bright field because for whatever biological reasons, we did not want to use a fluorescence stain in these particular samples. We wanted to keep them alive and not put anything toxic on them. So the goal of this assay is to count the cells and the area covered by the cells accurately using bright field only, or it could be done with phase contrast. And this will replace various traditional assays, including high content with a fluorescence stain, which in the past has been sometimes difficult to quantify using bright field only. Now, there are challenges when you're doing a bright field analysis. There are often artifacts such as scratches on your plate in the wells that you can see in the bright field images, and also uneven shading, which is more apparent in transmitted light, can make thresholding difficult. And this is even worse at low magnification. So this example we've got is at 4x, just to illustrate that. Okay, so here are some examples. I actually only have 3 different example images that we're going to be using here for this. So you'll see on the left is one image. This is a very densely populated well. The cells are confluent and very close together. We have another one that's sparse. And then finally another phenotype that shows some of these scratches. They're kind of shadowy because they're out of focus in the plane that we're looking at. And this is showing now the full image, so you can see also the shading effect. And the corners of the images are definitely darker than the center of the image, which is brighter, and you can see all of the scratches. And despite that, we're able to get a very accurate count of the cells that gives you consistent results across these different phenotypes. Okay, so the strategy that we use for doing these types of transmitted light images is to first apply some type of filter that enhances the signal of your objects or your cells. So we have a few different filters that you can choose from in the custom module editor. Next, you're going to use one of those Find Objects tools to find your cells. And then if you need to do any kind of filtering to remove clumps or other undesirable objects, you can add in a filter at this point. And then finally, you'll put in your measurements of interest. And you can see here how well it finds those cells, even with those scratches there. Okay, so I'm going to go ahead and go back to the software. And now I'm going to select my cell proliferation plate. These are CHO cells, again, read at 4x. And you can see the different phenotypes we have here. That's the dense one. That's the very sparse one. And this is the kind of intermediate with the scratches on. So I'm going to go ahead and go to my Run Analysis tab and click on Create Custom Module. Okay. All right. So here's my image. Let's zoom in a little bit so you can see. So the first thing that I need to do is to kind of enhance my objects in the image. And to do that, I'm going to use one of our image filters. And those are found under the Modify Image collection here. And you'll see a number of different tools here. There's arithmetic tools, a number of morphology tools, and some special filters available. So depending on your particular sample, your particular images, there's different filters that you might want to use. Some of the ones that we find useful for transmitted light images are the Gaussian filter and the LOG filter, which is the Laplacian of Gaussian. What I'm actually going to use for this particular example is the H basin, because all of these cells are quite small, and the H basin helps find some localized minima in the image. And all I have to do is select my source image, which is my transmitted light image, and then specify a threshold. Okay, so for basically the difference between the cells and the local background. So I can just mouse over a cell, and you'll see the intensity is about 36,000, and then it's about 41,000 in the background. So we can try different values. You want something that's going to really highlight them nicely. All right, so now you'll see on the right an image that's highlighted all those dark localized minima, and it makes this look more like a fluorescent image, which we know how to work with. And so you can see what happens when I try some different values here. If I set something much lower, I'm getting a lot of noise in the image, and if I set something that's too high, I'm going to not highlight them as clearly. So pretty simple to do that. So I'm just going to name my results here. Highlighted cells. And next I can just go ahead and again run one of my Find Objects tools. So I'll use the Find Blobs and start with my highlighted cells. And then I can again do the click to find and click on these cells. Oops. If I make a misclick, I can just hit delete and remove one. Okay. Let's see how well that did. I'm going to go to the split view because that makes it easier to see. And I can move back and forth. I can look in the corners of the images. And see. So I think my threshold is maybe a little bit high here. So I'm going to go ahead and lower that. Okay. I think that's finding it more accurately. I'm finding now those corner objects that I was missing with a higher threshold. So So if you aren't able to click on enough representative cells, you may have to manually adjust things like the intensity setting. I'm going to name my results cells. And then the last thing I might want to do is, just in general, sometimes we don't trust the objects that are right at the border, or we're not going to get accurate sizes because they're cut off. So optionally, you can add in this remove border objects. And this will just simply remove any objects that are touching the border of the image. Okay. You can see that side-by-side view, all the border objects are removed, and we'll just get true full cells here. Okay. So that's all I need to do for segmentation. I can go ahead now and set up my measurements. My mask will be my interior cells. My image to measure will be the transmitted light. And the two measurements of interest are my total cell area here. And my cell count. Okay. All right, so it went ahead and did it, and you see again that although you can see the scratches in the image, we're not picking them up because they were too large to be, and they were filtered out by that H-basin filter that we ran. Okay. All right. So I can go ahead and name this module. Okay. All right. And now I can run this analysis for the plate. Okay. There's my cell count total here. And if I click on any of the images, you can see the overlay, finding the cells nicely despite the shading, and it's avoiding the border objects. Okay, so that's transmitted light segmentation. I'm going to go back to my presentation now, and we'll move on to the last one, which is the cell migration assay. And so for this one, we use the ORIS cell migration assay, which is available from Platypus. And I'm sure some of you have tried this or at least looked into this. A nice setup for a migration assay. There's a plug that you put into the wells in your plate, and then you add your cells around the plug into the well, and you incubate them and allow the cells to seed. And in basically this outer ring around the plug. And when you remove the stopper, you'll have a hole in the middle of your well. And then you can incubate over time with that plug removed under different treatment conditions and see how much the cells migrate back into that hole. So the goal is to measure the area covered by those migrating cells, or the area of the hole the hole would be larger without migration, and the hole is smaller when there is migration. The main challenge in this assay is pipetting artifacts that are outside of the area of interest, which you'll see in some of the example images that we have. And the input that we're using is just a propylium iodide stain of a whole well image from the MetaXpress Micro. Okay, so this is just a montage of thumbnail images, and you can see, as I mentioned, some of the wells have these holes that are outside the central hole, and those are just pipetting little divots with the pipetter. That's what we're trying not to count. We just want to count the center hole and how much of it is blank and how much of it is covered by cells. And this plate had a few different treatments on it. Just to show you what the results were like, this measurement was the percentage of the whole area that was open. So the lower the number, the more the migration, I believe. Okay. All right, so we're going to start by thresholding the image. And then to only find the center hole, we're going to filter by the centroid of our object. And then we're just going to measure the area. So very simple analysis. Okay. So you can see as I click around, let me turn off this auto scaling. You can see the hole that we're looking for. Okay. So I'm going to go ahead and create a custom module. Okay. So the first thing I'm going to do is just a simple threshold to select by intensity. And just to save time, I'm just going to type in the values that I already know. Okay. So you can see that the dimmer objects are found, but I have a lot of false objects that I'm not interested in. So now I'm going to add in a filter mask. And I'm going to filter by centroid and by area. And again, just to save time, I'm going to type in values that I already know. So this is filtering by centroid Y, so you can see ones that are too high or too low get removed. I'm going to add in the centroid X. And that will remove objects that are too far to the left or the right. And just to be, make sure I've really found the right thing, I'm going to put in a max filter just in case there's some sort of artifact in the image. I have a centroid Y and centroid X filters for position and a maximum area filter, so I don't find anything that's larger than this hole is supposed to be. And then just so I can get a count across the whole image, I'm going to add in another simple threshold. And it should just give me one solid mask so that I can get measurements across the entire image. So my mask of objects is going to be the entire image, but I don't need any measurements from this. And then my feature group of interest will be the center hole. And for this, I want the total hole area and the count, just to make sure that I only get one hole per well. Go ahead and click apply. And now I get one object, one hole here, and that whole area. Okay, so I name and save my module, and then I can return to MetaXpress and run it on a few selected wells, and it runs very quickly, and you can see the total hole area there. Okay. So again, another very straightforward analysis using a custom module editor that would have taken quite a while with a journal to put together. I just wanted to show an overall a subset of some of the different custom modules that have now been created and are in use, just to give you an idea of the breadth of assays that we can cover with this custom module editor, far more than we can with any CAN modules. We're really excited about this because it really allows our users to develop new assays very quickly and get their experiments up and running much more quickly. So I think at this time we're ready to take questions. This is just a reminder of what Grisha showed at the beginning of how to submit your questions. So press on the Q&A button, type in your question, and ask all panelists. So please go ahead and submit any questions now.